Welcome to this short tutorial on apoptosis. Okay, since the topic is quite vast, for the sake of convenience, I have divided this into two parts. So, in this part one, let us understand what is the definition of apoptosis, and let us know what are the causes or conditions where apoptosis can be seen, and in detail, we will understand the mechanism of apoptosis. Okay, that is the extrinsic pathway and the intrinsic pathways of apoptosis, and the way the execution of apoptosis takes place. Okay, apoptosis. Now, how do you pronounce this? It could be pronounced as apoptosis or apoptosis. Actually, there is no consensus as to how to pronounce this as of now. Now, what is apoptosis? Apoptosis is a pathway of cell death, which is induced by a tightly regulated suicide program. So, what really happens here is that when the cells are destined to die by committing suicide, what they do is that they activate a series of enzymes within the cell, which actually degrade their own DNA material. Okay, and then proteins. This was first discovered in the year 1972 by John Kerr, Andrew Wyllie, and Alastair Currie, who were the great pathologists. So it's derived from the Greek word which means falling off, which means like leaves falling from a tree. See, the pattern of events in apoptosis is so orderly that it is often referred to as programmed cell death. So basically, we need to understand that apoptosis is a way a cell commits suicide. Now, why do a cell commit suicide? Why do you think a cell should commit suicide? One, it is very much important for the proper development. That means to say that apoptosis can occur under physiological conditions. Two, it is needed to destroy those cells which represent a threat to the integrity of the organism. So, apoptosis can occur in pathological conditions. Now, let us understand under what physiological conditions and under what pathological conditions apoptosis can be observed. The first and the foremost physiological condition is embryogenesis. The sculpting of human embryo is all because of apoptosis. Okay, so when I say that, I mean to say that, for example, the formation of fingers and toes of the fetus, it requires the removal of the tissue between these fingers and toes, and that happens by apoptosis. Okay, the second one is involution of hormone dependent tissues, for example, menstruation. See, the sloughing of the lining of uterus or the endometrium is because of apoptosis. The atrophy or atresia of follicles of ovary in the menopause is because of apoptosis. The regression of the breast parenchyma after lactation during weaning is because of apoptosis. The third instance where apoptosis can be observed under physiological conditions are loss of cells in proliferating cell populations. For example, the cells in the bone marrow, the cells in the thymus, the epithelial cells of intestine. These are continuously dividing cells. Okay, these cells just can't accumulate over a period of time. So, okay, these cells have to be knocked off to maintain a constant number. Okay, and that is what we know as homeostasis, isn't it? Now, the fourth instance is removal of self-reactive T lymphocytes. If these self-reactive lymphocytes are not knocked off, okay, you can expect an autoimmune response. So, the removal of these self-reactive lymphocytes is by the process called apoptosis. Lastly, death of inflammatory cells after their functions are over. What I mean to say here is that, for example, neutrophils are often recruited in acute inflammatory process. Once the function of neutrophils are over, then these cells, they sense that they are no longer needed and that they commit suicide. Okay, so these are the various physiological conditions where apoptosis is observed. So, what are those pathological conditions where apoptosis is found? One, the most important thing is that apoptosis occurs whenever there is an injury to the cell which cannot be repaired. The most common cause of such injury is DNA damage. Okay, so what happens here is that whenever there is a DNA damage, the cells respond to such DNA damage by the production of P53 gene. So we need to understand that P53 is the most important inducer of apoptosis. Okay, the second important entity where apoptosis can be observed under pathological conditions is that whenever there is a misfolded protein accumulation. So when I say misfolded protein accumulation, it means that there will be accumulation of proteins in the cell, okay, that has failed to fold properly into their proper tertiary structures. Okay, so these are the ones which these are the cells which can undergo apoptosis. The third one is apoptosis can be seen in cells which are infected by viruses. Okay, so the cytotoxic T lymphocytes, they kill virus-infected cells by the process called apoptosis. Sometimes in parenchymal organs after the duct obstruction, for example, in the case of pancreas, in the case of parotid, in the case of kidneys, you can expect some amount of apoptosis, and that can result in pathological atrophy. Now let us understand the pathogenesis of apoptosis. So how actually these cells commit suicide is what we're going to understand now. To understand the mechanism of apoptosis, let us see who is the most important player in helping the cell to commit suicide. The most important player are the proteases. Okay, so you know that proteases are the ones which actually cleave proteins. So these proteases are the ones which are aspartate directed. Aspartate directed means the proteases cleaves the proteins at the site of aspartic acid residues, and we should also know that these proteases are cysteine dependent. So the most important players are the cysteine dependent aspartate directed proteases. In short, they are referred to as caspases. Okay, so what are the types of caspases? So there are two major types of caspases. One is initiator caspases and two executioner caspases. Okay, initiator caspases are the ones which actually initiate the process of apoptosis. Executioner caspases are the ones which actually execute the process of apoptosis. So what are these initiator caspases? The caspases number 2, 8, 9 and 10 are the initiator caspases. The executioner caspases are caspases 3, 6 and 7. So do remember these initiator caspases which are 2, 8, 9 and 10 and executioner caspases which are 3, 6 and 7. Some basic understanding of these caspases. We should note that all caspases, whether they are initiator caspases or whether they are executioner caspases, they will be in the inactive form. These are known as procaspases. Okay, so the initiator caspases are activated by either of these two pathways. One is either by intrinsic pathway or by extrinsic pathway, which we'll be discussing in a short while. Okay, see, the executioner caspases are activated by the initiator caspases. Okay, now we know that apoptosis is basically a two-step process where the first step is called initiation and the second step is called execution. So the initiation is brought about by two pathways, extrinsic pathway and intrinsic pathway, which culminates into activation of executioner caspases. From this step, there is the process of execution. Okay, so what happens when there is activation of executioner caspases? The substrates are cleaved, and then you find the morphological features of apoptosis. So let us understand these concepts in more detail. Now, so we'll talk about initiation, extrinsic pathway and intrinsic pathway. Let us assume that this is a plasma membrane, a bilayered plasma membrane, and this is something called death receptor. Okay, so the death receptor which we are talking about is it could be either FAS or tumor necrosis factor receptor. Okay, so there is always a ligand which can bind to a death receptor, and these are known as death ligand. Okay, so the death ligand are FAS ligand. Since this particular step involves the death receptor and death ligand, it is also known as death receptor pathway or extrinsic pathway. So what happens in extrinsic pathway? We'll see now. So in this particular pathway, whenever the ligand binds to a death receptor, some conformational changes occur wherein more and more death receptors come and join together. Okay, so in this particular case, three death receptors join together and then they form a death domain in the cytoplasm of the cell. Okay, now after that, some adapter proteins come and attach to these death domain, and these are called FAS associated death domain or FAD. Okay, the basic function of this particular FAD or FAS associated death domain is activation of procaspase number 8. So we need to know that the caspase number 8 or procaspase number 8 are the initiator caspases. So FAD activates procaspase 8 and then that is converted to a activated caspase 8. This activated caspase 8 further activates activated caspase 3, 6 and 7. Okay. The caspase 8 which was in inactive form as a procaspase is now activated to form activated caspase 8. We know that caspase 8 is a initiator caspase. So this particular initiator caspase activates the executioner caspases. Okay, so what are those executioner caspases? These are caspase numbers 3, number 6 and 7. Okay, now these executioner caspases, they actually break the nucleus by activation of endonuclease, and two, they also break the cytoskeleton. Okay, so what happens when these executioner caspases are activated? They break the nucleus, they break the cytoskeleton. So let us understand this by this schematic diagram. Assume that this is a cell and there is breakdown of nucleus and cytoskeleton. For some unknown reasons, there is a formation of bleb. Nobody as of now know why bleb formation takes place. Okay, so these cytoplasmic blebs, no, later they are pinched off and then they are called apoptotic bodies. Okay, so we need to know that these apoptotic bodies are actually a membrane bound bodies which contains fragments of nucleus and then fragments of cytoskeleton. Okay, so apoptotic bodies are membrane bound structures. Okay, and since they are membrane bound structures, we now know that none of the contents are released outside. When the contents are not released outside, that means to say that there is no inflammatory reaction. So apoptosis, there will be no inflammatory reaction. Now, what happens to these apoptotic bodies? Normally, in the plasma membrane, uh, the phospholipid phosphatidylserine is found in the inner surface of the membrane. Whereas in these apoptotic bodies, this particular thing flips and then the phosphatidylserine is expressed outside. Okay, so now the receptor for this phosphatidylserine is found in the macrophages. So basically the phosphatidylserine is expressed outside and then now these becomes, sort of eat me type of bodies. So these are devoured by macrophages which have the receptors for those phosphatidylserine. So basically, these apoptotic bodies are phagocytosed by the macrophages. The process of phagocytosis is so efficient that these dead cells or these apoptotic bodies disappear often within minutes, without even leaving a trace. And there is absolutely no inflammation. That is the most important aspect of apoptosis. So this is what happens in extrinsic pathway. Okay, and then culminating into a common pathway, which is what we have described now. Now let us just quickly recap what happens in extrinsic pathway. So this is a death receptor pathway. We all know that they have a death receptor and the death domain. So some adapter proteins gets attached to these death domain, which have their own death domains, and these are called fast associated death domain. Okay, these are the ones which activate the caspase 8, and then finally the caspases 3, 6, and 7 are activated, which are executional caspases. Execution is basically by the nuclear breakdown and the breakdown of cytoskeleton, which forms apoptotic bodies, which are finally devoured by phagocytes. Okay, so this is the death receptor pathway with execution. The second most important pathway is called intrinsic pathway. Okay, so this is the pathway where mitochondria is involved, and that is why it is also referred to as mitochondrial pathway. Now, let us assume that this is a structure of mitochondria. Okay, these red dots or red circles, what have depicted here is these are cytochrome C. So one thing about cytochrome C is that as long as they are inside the mitochondria, normally the cytochrome C is located in the intermembranous space. Okay, as long as they are inside the mitochondria, they are really good. Okay, but once they come out to the mitochondria due to any reason, and they are the ones which actually activates apoptosis. Once these cytochrome C are released into cytoplasm, they combine with a protein called apoptosis activating factor 1. Okay, so the combination of these two results in the formation of an interesting structure called apoptosome. See, this apoptosome is actually a wheel-like hexamer. It's a complex structure. What this does is it activates procaspase 9. So procaspase 9 is converted to an activated caspase 9. Now we know that 8 and 9 are initiator caspases. Okay, so this activated caspase 9 activates caspases 3, 6, and 7. Okay, so these caspases 3, 6, and 7 are executioner caspases, and then the further steps are similar to what we have described. And these apoptotic bodies are engulfed or phagocytosed by macrophages. Now, what we have understood in intrinsic pathway is that the cytochrome C, which is normally present in the intermembranous area, is released into the cytoplasm. Now, let us understand how this cytochrome C comes out of mitochondria. Okay, so for this to understand, let us know. See, another important mechanism in intrinsic pathway is that there will be release of some proteins called SMAC, Diablo. These are the mitochondrial proteins. So what these do once they are released into the cytoplasm, so they actually neutralize something called IAPs. These are physiologic inhibitors of apoptosis. Okay, so normally the function of IAP is to inhibit apoptosis. But what happens when these are released? These released proteins they neutralize IAPs. So when these IAPs are neutralized, then there is a caspase cascade. Now let us understand how these cytochrome C, which was normally present within the mitochondria, comes out of mitochondria. Okay. For that we need to understand the regulation of apoptosis. See, the regulation of apoptosis is basically by BCL2 family of genes, which is located on chromosome number 18. Okay. So there are two important set of genes. One is anti-apoptotic genes and two pro-apoptotic genes. Anti-apoptotic genes are the ones which actually prevent apoptosis, whereas pro-apoptotic genes are the ones which favors apoptosis. So the anti-apoptotic genes are BCL2, BCLXL, and MCL1. The pro-apoptotic ones are BAX, BAC, BCLXS. Now, what normally happens here is that in any given cell, whenever there is a stimulation of these cells by the growth factors, okay, so the growth factors are the ones which actually activates these genes, the BCL2 genes, okay. So these BCL2 genes are present on the outer membrane of mitochondria. See, the function of these genes, BCL2, BCLXL, and MCL1, is basically to prevent the leakage of cytochrome C outside the mitochondria. Okay, so this is what happens normally. Now, the pro-apoptotic genes are activated whenever there is a cessation of growth factors. Okay, so what happens here is that these genes are activated, BAX, BAC, BCLXS. Once these genes are activated by cessation of growth factors, the genes which are BAX and BAC, what they do actually is they oligomerize the outer membrane. These proteins oligomerize with the outer mitochondrial membrane proteins, and then they promote the leakage of cytochrome C. Okay, now we know that the anti-apoptotic genes prevent the escape of cytochrome C, whereas pro-apoptotic genes encourages the escape of cytochrome C from the intermembranous space. Okay, apart from anti-apoptotic and pro-apoptotic genes, there are another set of genes called sensors. Okay, the sensors are BAD, BIM, BID, and PUMA. Okay, what these sensors do? These sensors inactivate anti-apoptotic genes, whereas they activate pro-apoptotic genes. So basically, sensors are also pro-apoptotic. Okay, these sensors are activated whenever there is a stress to a particular cell. Okay, so these sensors actually regulate the pro-apoptotic and anti-apoptotic genes, and that is the reason why they're also called as arbiters of apoptosis. So this is a summary of mechanism of apoptosis. Okay, so one is death receptor pathway and two intrinsic pathway. In the death receptor pathway, we know that there is a death domain, fast associated death domain, which activates procaspase 8, and then the executioner caspases are activated. In the mitochondrial pathway, the cytochrome C is released outside. They combine with apoptotic activating factor 1, forms apoptosome, and then the caspase 9 is activated, which in turn activates the executioner caspases. And now we know the execution is in the form of nuclear breakdown, and the cytoskeletal breakdown, formation of apoptotic bodies, and then they are phagocytosed by the macrophages. So this completes part one of apoptosis, where we described what is apoptosis, where we talked about the physiological and pathological conditions where apoptosis can be seen, and three, where we discussed the mechanisms of apoptosis, in particular the intrinsic and extrinsic pathways of apoptosis. Okay. In the part two of apoptosis, we will learn more about the differences between apoptosis and necrosis. We will learn how to diagnose apoptosis, and three, we will learn different types of or examples of apoptosis, and finally, we will know about what is known as pyroptosis and necroptosis. Okay, thank you.